right? We're going to cover most of the material in this chapter, not all of it, but we should be able to cover half of it today and the other half on Tuesday. So after I get done lecturing, we'll figure out when we're going to take the test, right? Because this is the end of the material for this module. And sometime today I'll try and post the Connect assignment for Chapter 10, which is only Section 5. So for today, this chapter focuses on DNA replication. And I think a few times before I've said some things as relates to this, and I don't know your guys' background knowledge, so hopefully we'll fill in the gaps. So obviously this is how we copy, right? Most of us are familiar with the fact that we copy our DNA and how basically it's done. So we're going to go into some of the specifics. And it actually occurs really quickly in our cells. I don't remember if they give any time frames in this chapter. I don't remember reading any, but I've seen some in the literature. So we use, it's double-stranded, right? So we use one strand as a template to make the new strand. But that wasn't always known, right? So we'll talk about the features that make it easy to replicate, the structural features of the DNA. So we've gone over quite extensively the structure of the DNA, so this just kind of piggybacks on that. And then we're going to analyze and look at the experiment, and there's actually an animation that comes with your book. So again, I think what I'll do for you guys, sometimes what I've done in the past is I've put these animations into the PowerPoint, but right now I have them in the separate PowerPoint. I think I'm going to do like I did for Chapter 9, I think it was. I'm just going to post that separate PowerPoint if you guys want. But remember, these are in your book, right? You can click on them in your book, but sometimes it's just easier to open up the PowerPoint. The important thing there is that you have to be in presentation mode, otherwise it doesn't play or work. You don't have to be on the internet, because I was watching them this morning before my doctor's appointment, so and I didn't have internet. So that's good news as far as that goes. So for DNA, right, we already know Chargaff, got lovely names, rule, right, that A with T and G with C, right? So this complementary binding that happens between the two strands. So the new strands are commonly referred to as the daughter strands. The original strands are referred to as the parent strands. So the parent strands are actually the template for the new strands, which we call the daughter strands. So we take a strand, right? We're going to separate the two strands, and you'll see that this one's coded in purple here for the parent strand or the template strand, and then the new strand is coded here in blue. You can see the sugar phosphate background is coded in blue. So this happens for both strands, and then we end up with two copies. Somebody above us, apparently. No. Oh, it might be. Here, I'll just mute it. Yeah, that's what it is. That's what it is. I heard the bang, not the, yeah, it's background noise. I'll just mute it for now, but I will turn it on. Actually, I'll plug this in, because there's a couple of animations I want to show you guys. So in the 1950s, right, we knew the structure of the DNA, but trying to figure out how it actually copied itself, right, how it became replicated. There was three theories, actually, where both parent strains stayed together after the DNA replication. What actually happens, which is the semi-conservative model, where one parent strand and one daughter strand make up the new strands. And then this, I have to say, is the strangest one. The dispersive model, where they believe that it was different pieces of parent and daughter one. Like, how would you put that together correctly, right? It just doesn't seem to make sense to me why they would think it was like that. So these are just diagrams, you know, showing what they believe the possibilities were, right? that the parent strands ended up coming back together potentially, right, and the daughter strands together, instead of what we know actually happens now, where one strand of the parent strand and daughter strand make up one of the newer strands because the two parents separate and became the template for the daughter strands. And then this is the dispersive model, right, where there's different pieces of daughter and parent one. So, Meselson, Matthew, and Stahl in 1958 did this really cool experiment. So again, we're kind of labeling stuff. In this case, they labeled the nitrogen component, right? So we know the nitrogenous bases contain nitrogen. So they labeled that with a heavier isotope. It tends to have more, what is it? Neutrons or protons or both? I can't remember. I should know that. Yeah, but I forget what makes it heavier. I think it might be the neutrons, right? So N15, right, is a heavier form of the nitrogen. And so there's actually an animation for this that I want to show you guys because I find it's pretty cool. I don't know, I'm a visual learner. I don't know about you guys, but when I see things in motion and pictures, it helps. So that's this one. So we'll go ahead and play this one, then we're going to talk about it. But that's that, and they explain that here. Again, they don't go down to the molecular level on how it's heavier. They just say it's heavier. So obviously we don't need to know that for this class, but as a scientist we should. Yep, I'm going to turn it back on. Thank you. All right, so hopefully this works, but if not, I'll restart it.
The Meselson and Stahl experiment provides evidence for semiconservative replication of the DNA molecule, where the two parent strands serve as the template for synthesis of new strands. In this experiment, bacterial cells were grown for several generations on a medium containing a heavy isotope of nitrogen, N15. The DNA in these cells, therefore, contains N15, nitrogen. The cells were then transferred to a new medium containing the normal lighter isotope, N14. At various times after the transfer, samples of the bacteria were collected. The DNA was then extracted and dissolved in a solution of cesium chloride. The samples were then spun rapidly in a centrifuge. When the cesium chloride is centrifuged at high speed, a concentration gradient is established in the tube. DNA molecules move in the gradient until they reach a place where their density equals that of the cesium. DNA containing N14 moves to a position in the gradient determined by its density. DNA containing N15 is denser than that containing N14, so it sank to a lower position in the cesium gradient. After one generation in N14 medium, the bacteria yielded a single band of DNA with a density between that of N14 DNA and N15 DNA, indicating that only one strand of each duplex contained N15. After two generations in N14 medium, two bands were obtained, one of intermediate density, in which one of the strands contained N15, and one of low density, in which neither strand contained N15. Meselson and Stahl concluded that replication of the DNA duplex involves building new molecules by separating parent strands and then adding new nucleotides to form the complementary strand on each of these templates.
And I like this diagram. This is similar to what we just saw, right, when we looked at the conservative. So they've color-coded it. So the green is the heavy, right, and they took it out of that environment. So all the DNA was that heavy variety. And then they put it in new media where when they make up the new stuff, it was the lighter stuff, right? And so they were able to conclude that because what happened when they looked in the tube, right, it was a mixture of heavy and light, so it settled there in the middle between the two. So simple, simplistically elegant, right? It's beautiful. But then as we go, right, those two strands separate from each other, right? And they're still in that media where it has the light. So that's why in this last one, we end up with light ones, right? If they then put it back on media with heavy, then we'd end up with a heavy band, right? So we have the ones that are heavy and light, right? And then they're going to separate. And so therefore, right, so this is showing one cell here at the top and another cell down at the bottom. Does that make sense to you guys? Right? So in the new cells, they're going to have the heavy and the light. So they end up with a mixture and then one that is all of the new light nitrogen. Make sense? Okay. All right. So back to over here. So they did this, several times. So, after several generations, right, and they looked at the different bands, right, as it settled out in that medium. Here we go. So here's that mixture of, it's hard to see. I wish I would have drawn the lines all the way across, huh? But you can see that very top band is light, right? The middle there is halfway between, and then the heavy here at the bottom. And it goes in this direction, right? So we have to start this process, right? DNA replication's got to start somewhere. So for bacteria, they have we talked about this last time, usually a single circular piece of DNA that makes up their chromosome. Because of that, they just have, and they tend to have less DNA than we do in comparison. They have a single site, right, where replication begins, right? It's referred to as the origin of replication, right? A lot of times abbreviated ORI. And so for replication to start, to separate those two strands from each other, so the proteins can come in and actually copy the DNA, there's an important group of proteins known as the DNA, this one's hard to say because you have to say A again, DnaA proteins, right? Or it's DnaA protein, but several of them interact with the DNA. And so I'm going to flip back again because this one's super cool, and this one will really help you guys with studying. I don't know why they did that this time and didn't do it with any of the others. This is it. So not going to play because I watched it earlier. There we go. Let's see if we can reset it. You think I have to bring it back? Yeah, you're probably right. Thank you.
Begins at a region of the bacterial chromosome called the origin of replication. The origin consists of an AT-rich region adjacent to sequences that are recognized by the DNA binding protein, DnaA. The binding of DnaA to the DnaA boxes initiates a process that leads to an opening of the AT-rich region.
It's not doing it.
Bubble.
Is it because I already went through it once before? Okay, I'm going to close this out. I'm trying to get this to reset itself. It's not doing what I wanted it to do. That's not what I want. I want the animations. That's it. Let's see.
The process of DNA replication in E. coli begins at a region of the bacterial chromosome called the origin of replication. The origin consists of an AT-rich region adjacent to sequences that are recognized by the DNA binding protein, DnaA.
See, the questions pop up now. Those little dots questions pop up. So this is good because it, right, gets us thinking along the way. So this one, you guys already know the answer to it because, right, it was played it through before. Let's see if you were paying attention, right? So what happens next? So we have this origin of replication area, right? We have DnaA protein, right? Does the DnaA proteins separate the DNA in the region of the DNA boxes? The DNA proteins bind to the DNA boxes, causing the ATC-rich region to separate. Helicase proteins bind to the DNA and create replication forks, or DNA polymerase binds to the DNA and starts making the leading strand. Right, this is pretty self-explanatory, right? This DnaA protein, or several of them, are going to bind to these regions. This is why they're called this, the DnaA boxes in the DNA, right? In doing so, it's going to create a conformational change that you'll see in a moment that helps separate the AT-rich region. Why do you think that's an AT-rich region as opposed to a GC-rich region? Okay. No, not start.
It has to do with the bonding.
It has to do with the bonding, right? So what do we know about the bonding between A's and T's versus the bonding between G's and C's?
Not as strong.
It's not as strong between A's and T's.
It's exclusive to DNA only, not RNA.
T's are, yes. But there's a reason why we're talking about the
It's a
AT
double
bond.
bond, not a triple bond.
Right. This is important, and this is why I try to tease it from you guys, because you got to know it, right? So A's and T's, there's 2 hydrogen bonds, right, that bond them. This is very important. Between the G's and the C's, there are 3. So it makes sense that in this region that comes apart, there's a lot of A's and T's, because that's a less stronger bond, right? You only have to break 2 bonds as opposed to having to break 3 bonds. Make sense? So remember I said when we want to separate DNA, like scientists take that into consideration, because it's going to take more energy if you want to separate a code that has a lot of G's and C's as opposed to a code that has a lot of A's and T's. What we refer to as AT-rich, meaning a lot of A's and T's. Okay? All right.
The binding of DnaA to the DnaA boxes initiates a process that leads to an opening of the AT-rich region.
Right, so it makes sense that that's AT-rich, right? That's easier to pull apart than G's and C's. So what's going to happen next, right? So we've separated the 2 pieces. So it says topoisomerase leaves positive supercoiling. Primase makes DNA primers at the origin. Helicase proteins bind to the DNA and create the replication forks. DNA polymerase binds to the DNA and starts making the leading strand. So we've got an opening right now, right, between the 2 strands. What's the next thing that you think we need to do? Yeah, we got to keep that opening, right? We've got to keep them, and we want to literally separate the 2 strands, and that's referred to as the replication fork. And look, if you get it wrong, it'll give you another try, by the way.
The replication bubble in the DNA molecule is opened further by helicase, and the replication forks at each end of the bubble are expanded.
So it's going to go in 2 directions, right? Because we have 2 strands, right? It's going to go in 2 directions. So now what? What protein is needed to prevent? So see how we've got them separated? How do we keep them from binding back to each other? What's going to help us do that? Topoisomerase, primase, DNA ligase, or single-strand binding protein?
DNA polymerase?
How are we going to keep the 2 strands, right, from binding back on each other? Yeah, because it sounds like single-stranded. We want to keep it single-stranded, right? It's a no-brainer, y'all. A lot of these make sense, right? So we want to keep a single-stranded. How do we do that? We have proteins that bind to the single-stranded DNA to keep it from binding back on itself. And they're called, very simply, single-stranded binding proteins.
Single-strand binding proteins then attach to each single-stranded DNA molecule to stabilize the open bubble, and subsequently topoisomerases bind to relieve the supercoiling of the double-stranded DNA created by the unwinding of the strands.
So some of you guys have probably experienced this too, like if you've ever done any type of knitting or cross stitch or anything like that, when you go to try and take thread or yarn that's been winded together and you're trying to unwind it, right? Sometimes it'll kind of bunch up if you don't pull on the other end, right? If you don't have some, I was going to bring some thread in today, but I have no idea where it is. I haven't unpacked that yet since moving this. I wasn't going to go buy new stuff when I have stuff somewhere in my house, right? But most of us have probably experienced that. Tyler, maybe not, right? I'll bring some in for next week. It'll be my mission to bring some thread so we can separate it, and you guys can see what happens when you try to separate it. So it'll kind of bunch up, and so because it's twisted around itself. So what the topoisomerase does is it helps relieve that twisting, right? So those enzymes you can see are binding here. So what enzyme is needed to initiate the synthesis of the strands? Can we just jump right in? What's the one that we're going to need next? Primase, right? It primes the system. And what does it actually make? Anyone know? It doesn't actually make DNA. The primers are made out of RNA. Primers are made out of RNA. So primase makes the primers, and that's the next step.
RNA primers are synthesized by the enzyme called DNA primase. To make the two leading strands, single primers are synthesized at the origin.
Okay, so they're going to make that. Which strand is made as one continuous strand in the same direction as the moving replication fork? The leading, the lagging, neither, or both. So they said which one, right? Right there it tells you it's one of them, right, y'all? Right, it is the leading, right? As the name implies, it leads, right? It leads the way. So it goes along.
DNA polymerase 3 then extends each primer by synthesizing a daughter strand of DNA in the 5' to 3' direction as it moves toward the replication fork. Each lagging strand is made in the direction away from the replication fork. DNA primase synthesizes short RNA primers, and then DNA polymerase extends the primers by synthesizing a short segment of the daughter strand called an Okazaki fragment in the 5' to 3' direction until it reaches the RNA primer from the previously made Okazaki fragment.
So the problem with that is, right, this enzyme can only go in one direction. But remember, DNA is anti-parallel, right? So the opposite strand is in the opposite direction. So as we're separating them, that leading strand, we can just go right down that strand. With the other strand, as it's being separated, it can't follow because it's got to go in the other direction. So as each little piece comes apart, right, it can add those segments. But the thing is, it can't just start on its own. It needs those primers. So it adds lots of primers, right, and little short segments of DNA. And of course, they're named after Okazaki, who helped discover this is what happens, right, for the lagging strand. So which enzyme is needed to remove the DNA primers, do you think? And it's actually going to make DNA. It's going to replace those primers with DNA. So there's a clue there for you. Which polymerase do you guys remember started it? DNA polymerase what? 3. So it's not that one, right? It's a different one. It's 1. Yeah. The first one they discovered, actually.
DNA polymerase 1 removes the primer and fills in the vacant region with DNA.
I didn't expect you guys to know that one. Right. So now it's, right, DNA polymerase 1 is gone and replaced those primers with DNA. But the problem is, you see these gaps between the Okazaki fragments? We gotta bind those nucleotides to each other. That phosphodiester bond, we gotta make that. So who do you think makes that? Topoisomerase, primase, DNA polymerase, or DNA ligase? Who have we not talked about yet? Right, and its name again tells you what it does. It ligates. It puts the pieces of DNA together.
Finally, 2 adjacent Okazaki fragments are ligated together by DNA ligase.
Pretty, right? All right. So I like that because it kind of, steps you through it and gets you to think at each one of the things. But apparently, when you open this PowerPoint, you got to reopen it again to reset it. So I had already played it this morning, and I was like, "Wow, this one is cool." Okay, so this is a diagram showing the replication fork. So there are 2 of them, right? Because it's going to go off in opposite directions from the origin of replication. And then, of course, we're going to talk about where it ends. So as we saw, right, the origin of replication for E. coli, it's ORI and C for chromosome. We saw that there's an ATC-rich region, and it makes sense why that's there, right? Because that's easier to separate. We have the DnaA boxes. Those are the sequences that the DnaA proteins actually bind to, which is going to help create that distortion in the DNA so that we can separate that ATC-rich region. In addition, there are GATC, G-A-T-C, methylated sites within this region, and actually along the DNA itself. So here's our ATC rich. You see there's a lot more A's and T's than G's and C's. Here are those repetitive sequences, right, that the DNA binds to, the DnaA proteins. And then there are segments where you see this sequence, GATC, and it becomes methylated. And do you guys remember why we methylate our DNA? Yeah, it's between the parent and the daughter strand. So our DNA becomes methylated, and so we know that that is the older DNA in the cell. And this replication won't happen without the methylation. So they know that they can't go there yet, right? So replication, one cycle starts, and it's not until those separate and that both strands become methylated that replication could potentially start again, right? Because think about this, if we started replication and then started it again, we'd have a big tangled up mess, right? So that's one of their ways of regulating replication, is methylation, right, of those sequences. So here's like what we saw in the animation, right? But it's cooler when you see stuff moving, I think so, right? So here's that binding and that pull and distortion on the DNA it creates by those DnaA proteins binding and popping open that region of ATC, AT-rich region, right? So the next thing that's going to happen is helicase is going to bind, right, to each one of the strands for the replication, and it's going to go in two opposite directions. So I already talked about the methylation, right? And so as I said, that's to regulate replication. So it won't start another set of replication until it finishes the first. So helicase, what does it do, right? So we already know that separates the two strands. It basically creates the forks and continues the replication forks. What does the topoisomerase do? It helps with the supercoiling, right? As it's being unwound, it keeps it from tangling up, basically, in front of the replication fork. Remember, the single-stranded binding proteins do just that. They bind to the DNA to keep it what? Single-stranded, keep it from not binding back to itself as we're doing the replication. So those guys are really important, right, for the proper unwinding of the DNA. Then we get into primase, right? And primase, what did that do? What did primase do? Primed it by making what? RNA primers, right? That's why they colored it differently too, by making RNA primers. DNA polymerase, as the name says, copies the DNA, right? Actually puts the nucleotides together on the strand. Which one acts first? DNA polymerase 3 or 1? 3. 3 is the one that actually generates the leading strand the whole way, right? And makes the Okazaki fragments for the lagging strand, right? So you're like, why is it 3? The numbers are in the order in which these were discovered, right? So 1 was actually discovered before 3. But 3 actually acts before 1, right? So the numbers aren't order, unfortunately, right? Numbers have to do in the order in which they were discovered. So actually 3 acts first. And then what puts together the pieces after the DNA polymerase 1 replaces the primers in the lagging strand? What is going to put those Okazaki fragments, link them together? The DNA ligase. Right. So what's the similarities and what are the differences, right? This is one of the things we're going to look into next between the leading versus the lagging strands. So we've gone over all of this. I don't need to read these slides to us. We've seen this in action. So there are actually 5 different polymerases they've found in bacteria. 1 and 3, as we know, is involved in normal replication that we just talked about. When cells have been damaged and when they're going through DNA repair, the other polymerases, 2, 4, and 5, they come into play under those circumstances. In trying to replicate the DNA or repair the damaged DNA. So polymerase 3 is actually made of 10 different subunits. So it's a really large protein complex that actually makes up that whole enzyme. Where DNA polymerase 1 is a single polypeptide, right? So a whole big long string of amino acids that folds back on itself and forms its globular structure. Let's see. Primers, leading and lagging. So leave it right there for that. Okay. So I'm going to play one of the other animations that shows leading versus lagging in a little bit more detail. I'm trying to remember which one it is. It's one of these two. You would think that these were the same, but they're different. Let me see. I don't think that's it. This one's boring. Well, I mean, it just goes like this. Zoop. I don't know how helpful that one is. Okay, so I think this is the one. Let me check. Yeah, this is the one. Okay. So let's watch it in action.
DNA replication begins when helicase unwinds a segment of the DNA and breaks the hydrogen bonds between the two complementary strands of DNA. DNA polymerase can only add new nucleotides to a free 3' end of a growing chain. Synthesis of one strand of the DNA, called the leading strand, proceeds continuously in the 5' to 3' direction. Synthesis of the complementary strand, called the lagging strand, is more complex. DNA polymerase can add new deoxyribonucleotides only to a free 3' OH. To provide a free 3' OH starting point on the lagging strand, RNA primase attaches to the DNA and synthesizes a short RNA primer. DNA polymerase 3 then adds deoxyribonucleotides to the 3' end of the RNA primer. DNA polymerase 1 replaces DNA polymerase 3, removes the RNA, and replaces it with DNA. Finally, the enzyme DNA Ligase forms a phosphodiester bond between the 3' OH of the growing strand and the 5' phosphate in front of it. During DNA replication, the leading strand is synthesized continuously, while the lagging strand is synthesized discontinuously.
Oh, powered would be good. So it's continuous, right? It only needs one primer for the which strand? The leading strand. The leading strand, they put down one primer at the site of the origin of replication, right? And then it just can go and follow as helicase is separating the two strands, right? So one primer versus the leading strand, the difference is, leading strand, one primer, lagging strand, I don't even know how many, right? But numerous primers, right? Depending on how long the DNA is. Okay. Why is it that the leading strand the polymerase can just start at origin and just follow that fork as it goes, as opposed to the lagging strand has to do these short segments? Right, 5' to 3'. So unfortunately, the enzymes can only go in that direction, 5' to 3'. So as we know with the structure of the DNA, it's anti-parallel. Why? I don't know. Right? But it is. And because of that, if you're separating this one, it needs to travel in this direction. So he has to keep hopping, right, and going back based on as the stuff becomes available to be copied. Okay? Why not just wait till the end? Jeez. Right? It would get all tangled up, right? So it's probably why it doesn't. So how does it solve this problem? Numerous primers, right, and small segments, and it works its way, right? So it's behind the other one all the time. So what's the same, though? So there's lots of differences, right? The direction is different, right? You need one primer for leading. You need multiple primers for lagging. You need extra enzymes even for the lagging strand, right? The lagging strand needs DNA ligase to put the Okazaki fragments together. You need DNA polymerase 1 to remove all those primers and turn them into DNA. What do these guys even have that's similar? Both the template strands are methylated. Yep. The direction in which the enzymes move, right, is 5' to 3'. They move in the same direction. And we still get A's and G's and T's with C's, right? But there's a lot more differences than there is similarities, for sure, between what happens from making these two strands.
DNA replication begins
I don't want
when
to
the enzyme helicase
play that one. That one's very similar to the next one.
Can't happen during DNA replication.
We're done with those. End. Okay. So here's where it's separating, right, from the origin. So we have the leading versus the lagging strands, right, made in the different segments on both sides of the replication fork. We're supposed to cover termination today? I guess we are. Is this all of them? Did I not delete the rest of it?
That was it. I'm looking through your file for the animation thing. It was
Yeah.
part 2 for chapter 11.
Yeah. I'm trying to remember what else I wanted to. Oh, this today. Okay. This is the confusing one for me. I don't know about for you guys. Okay. So the way they drew this out, the leading and the lagging, right? I don't know, space-wise or, explanation-wise, but they don't believe that that's exactly how it's arranged in three-dimensional space. So what they've found from different studies that they've done is that the helicase, right, and what does the helicase do?
Separates them from the air pocket hole.
Right, it separates the two strands. It breaks the hydrogen bonds between the two strands and separates them. What does the primase do? Puts the RNA primers in, right? And especially for the leading strand, it just does it once, right? One primer, and then it just goes, right? DNA polymerase 3 just goes from that point forward. For the lagging strand, the primase is going to have to keep laying down primers, right? As that strand becomes available. These two actually bind with each other. And that bound complex is referred to as the primosome. Then we go one step further. 2, right? So notice 2 DNA polymerase whole enzymes. So this is DNA polymerase 3, right? Which is actually multiple proteins that form this whole big complex known as DNA polymerase 3. 2 of them bind with the helicase and the primase. This is referred to as the replicosome. Replisome. Because that's where replication happens, right? So they have, so what's a replisome? What do we say it is? It's 2 DNA polymerases, specifically DNA polymerase 3. What other enzyme? 2 more. What separates the DNA? Helicase, right? What puts down the primers? Primase, right? So, where's our picture for this? There it is. Okay. So, this is the primosome. So this is the primase and the DNA helicase. So you see how it's kind of like a tunnel? They believe that one strand and that's the lagging strand, actually goes through the helicase and through the primase, because it's going to have to continually add primers. The leading strand actually goes outside, right? We just have to add that one primer at the beginning, and then the DNA polymerase is going to copy that. So there's your primosome. It's a primase and a helicase. And then attached to that are two DNA polymerases, one for the leading strand and one for the lagging strand. This whole complex is known as the replisome, right? Because it's where replication happens. So for that lagging strand, it loops around. And it actually comes, so I'm trying to say, it starts at each one of the primers, and then it goes to the next primer. So see here, it went from this primer, it's copying, and it's going to stop at this one. So then guess what happens? This has been spooling out while this has been copying. And then it's going to jump to this next primer. And then it's going to copy from this primer, right, back to this primer. Does that make sense to you guys? So I remembered years ago seeing this cool animation video on YouTube, and I found it this morning, right, that shows this, where you can see the looping and then the looping and the looping. Right? It shows a bunch of other stuff in the animation, but here I'm going to add, what did I do? I don't know what I did. I'm going to add, oh, I was hitting the wrong thing. I want new slide. Which one is, oh, right there. I was hitting the wrong spot. Okay. So, spell that right? Replisome. Replisome. Right, okay. So, I sent it to myself. And this isn't it. But that's okay, because I'm going to pause this, because this is one I want to show you guys too. I ran across it looking for this one. So this is kind of cool. I don't know how accurate this first part is, but I'll go ahead and play it for you because it kind of reviews some of the stuff we talked about in the previous chapter. Where is the linky thing? Okay, I think it automatically did it for me. So let's make this big. The resolution sucks. It's kind of blurry, but, you know. It's good enough.
You will see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then looped and coiled yet again. To the familiar shapes known as chromosomes, which can be seen in the nucleus of dividing cells. Chromosomes are not always present. revolve around the time cells divide when the two copies of the cell's DNA need to be separated. Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spinning off to the right.
Right here. That's
Things are
the
not
leading.
so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
It's kind of cool, huh? But you have to kind of orient yourself. They do a kind of a good job of letting you know what's what. So I'm gonna let me figure out how to mute her. Probably have to. There we go. All right. So let's make it big again. I wish it wouldn't go as fast as it goes. Right? It goes kind of fast. Okay. So here. I want to turn off the closed captioning too. Okay. So let's rewind a little bit. Okay. So that's the helicase, right? So as I said, see how that one goes on the outside, that strand? Here's the DNA polymerase, and that's the leading strand here, right? Leading off here. This is the lagging strand. So notice it's being separated, and part of it, right, goes, the primase is adding the primers there. You see that right there? And then DNA polymerase comes in and grabs it once it's finished that section, right? So it has to keep hopping. And it looks like what they believe is happening is that a new DNA polymerase comes in. Do you see? Comes into this complex and grabs onto the next segment and starts the next segment. And see, once it gets to the end, it pops off and another one pops in. Right? So then it just keeps going and going and going. So although they say, right, that they're all bound together, the one for the lagging strand has to keep popping off, right? Some theories believe that that's what's happening. So this just goes into transcription, what we'll get to later. But I find that was, I probably should try and find the original source of this video so it's not so grainy. I can't read that. W-E, I wish I could read that. W-E, I don't know. I have to do some research, some searching, yeah. But I'd see, huh?
Show more over there under the comments.
Show more. Yeah, I think it's the same though. DNA replication. Yeah, I don't know. Had to look into it. But I'd seen that, a student had pointed me to that one before. And I think it really kind of, shows that whole looping effect. It's probably one of the better ones I've seen for showing that effect that happens. So I'll save that one there. So while I was there, right, I had paused this other one. The reason why I had saved this link when I looked at this one is, here, let me see. Look at that. Isn't that beautiful? I just have to say that it looks so beautiful. But remember when we talked about the structure of the DNA and I was really kind of frustrated It doesn't look quite as good up there for you guys as it does for me. Can you turn off the front row of lights? One of those switches is the front row of lights. I forget about all this brightness. Okay, so then that's the other one. There we go. We should turn that off when I show you guys videos. So you can see. So you can see better detail now, huh? I don't know how that other one was for you guys. Was it too dark or you could see? You can see, okay. But I put the link for you so you can watch it again, over and over. But remember when we talked about the structure of the DNA? Is there a feature here that you think you can probably see a little bit better than we've seen in some of the other diagrams? So if you were to talk me through this, what are you seeing? What parts do you think you see and can identify in that double helix?
Adenine, thymine, guanine, and cytosine.
Yeah, and where are those? They're in the middle. So they're in here, right? And so what is this kind of blue-like looking structure?
Peptide bonds.
Close. Phosphodiester. So this is the sugar phosphate backbone right here. Now follow this along. Watch. I'm going to follow this one thread. Do you guys notice something?
One shade is darker than the other, showing the different bonds.
Do you see how between the sugar phosphate backbone right here, you see this gap? Do you see the gap here? Do you see how it's bigger here? Right? And it's smaller here? What do we call these gaps? No. The major and minor grooves. Right? And so there's more of the code accessible here in the major groove. So proteins that actually interact with code interact usually in the major grooves of the DNA versus the minor codes where proteins will interact but not usually directly with the sequence. Does that make sense to you guys? So what do you think the DnaA boxes are in? Minor or major grooves? So the DnaA boxes that the DnaA proteins bind to, are they a major groove or a minor groove? A major groove, because they're actually interacting with sequence. All right, so that's why I wanted to, this is a pretty good, I started to watch some of this video. This one's pretty good.
Do you recognize this molecule? This is DNA, or deoxyribonucleic acid. By the end of this
You can really see the difference there, huh?
The structural features of DNA, as well as describe the importance of those features for function. During this video, we will look at different representations of the DNA molecule to better view certain details.
I'm not going to play the whole thing.
Same structure. Inside the cell, you will most commonly find double-stranded DNA, in which two strands intertwine to form a double helix. The most common form of the DNA double helix, which is what we will discuss here, is also called B-form DNA. Now, let's move to a more simplified representation of DNA to discuss the details. We can unwind the double helix like this, so that we can see the chemical structure inside. Each strand is a polynucleotide, meaning the strand is made up of many individual units called nucleotides. A nucleotide has three components: the 5-carbon sugar, a phosphate group, and one of 4 possible nitrogenous bases: adenine, guanine, thymine, and cytosine. The nitrogenous base is always attached at the 1' carbon of the sugar. If we count from there, we can see that there is a phosphate between the 5' carbon of one sugar and the 3' carbon of the neighboring sugar. The sugar is called deoxyribose because it is missing a hydroxyl group at the 2' carbon, which is present in ribose. Because of this, nucleotides in DNA, deoxyribonucleic acid, are called deoxynucleotides. Nucleotides attach to each other in the DNA strand by phosphodiester bonds. The phosphate group of one nucleotide binds to the 3' oxygen of the neighboring nucleotide. Thus, we can
that the sugars and phosphate groups make up the DNA backbone. The carbon numbering is key to describing the directionality of the DNA strand, 5' to 3'. Looking within the sugars, there is an intrinsic orientation difference between the two strands. On the top strand, you can see that the 5' carbon of each sugar is on the left, and the 3' carbon is on the right. The opposite is true for the bottom strand. Reading left to right, that makes the top strand orientation 5' to 3', and the bottom strand orientation 3' to 5'. These strands are also sometimes called Watson and Crick. Keep in mind that this double-stranded DNA is still a double helix, and we have simplified the representation by flattening and unwinding the helix here to better see the atomic structure. Although the nucleotides come together through covalent bonds in the backbone, the two DNA strands interact through non-covalent hydrogen bonds between the bases. Each base forms multiple hydrogen bonds with its complementary base on the opposite strand. Bound together by hydrogen bonds, each unit is called a base pair. The hydrogen bonding contributes to the specificity of base pairing. Thymine preferentially pairs with adenine through 2 hydrogen bonds, and cytosine preferentially pairs with guanine through 3 hydrogen bonds. Thymine and cytosine are called pyrimidines, characterized by their single ring structure, and adenine and guanine are called purines, which have double rings. The geometry of the AT, or TA, and GC, or CG base pairs is the same, allowing for symmetry and base stacking in the helix. This mostly has to do with the distance between the backbones and the angles to which the base is attached to the backbone. Other base pairs, like GT for example, do not have the same geometry, cannot form strong hydrogen bonds, and disturb the helix. The double helix structure of DNA is highly regular. Each turn of the helix might have approximately 10 base pairs. In addition to the hydrogen bonding between the bases, the stacking of the bases also stabilizes the double helix structure. These pi-pi interactions form when the aromatic rings of the bases stack next to each other and share electron probabilities. The regularity of the helical structure forms 2 repeating and alternating spaces called the major and minor grooves. These grooves act as base pair recognition and binding sites for proteins. The major groove contains base pair specific information, while the minor groove is largely base pair non-specific. This is because of the patterns of hydrogen bond acceptors and donors that proteins can interact with in the groups. In this way, the DNA can be acted upon in either a sequence-specific or non-sequence-specific manner, allowing proteins to position themselves correctly in the genome to carry out their designated tasks. This is the DNA double helix, and you now learn the structural features that influence its function. We hope you've enjoyed exploring this amazing molecule test.
I have to say, that one is pretty darn good, isn't it? I'm gonna just go ahead and paste that one here in as well. Let me see. Let me make sure that's the right one. Yeah. I'll paste that one in for you guys as well.
This is beautiful, isn't it?
This guy is hysterical. If you've watched him, we don't have time for that. He's good too. Alright. So, we'll just write another cool video. I have to say that one, the detail is beautiful in it. That's all major stuff you need to know for this class. That's a good review one. Good review. Okay, I think the last thing that I want to briefly talk about is how it terminates. So for E. coli and bacteria, right, there are sequences and regions for each of the replication forks going in each of the directions that helps terminate the sequence. So of course they give them names called termination sequences, abbreviated TER. And there's 1 and 2, right, for each one of the 2 directions. And there are proteins that actually associate with those sequences that are going to help stop the replication forks from progressing past that point, right? So they don't run into each other, right? So that diagram is showing, right? So there's the proteins bound to those regions where those sequences are. And so one of them is for the strand coming from this direction, the other's for the other direction. And then you actually end up with, they're actually interlaced within each other, the 2 strands. And so they've got to be separated from each other. And so the enzymes that do that are the topoisomerases, right? Similar to the ones that untwist, right? The super coiling that happens from helicase untwisting. And then we got 2 separate pieces. For eukaryotes, right, for our linear chromosomes a little bit different. And that's what we're going to look at next time, right? We're going to look at the differences in replication for ours linear chromosomes. So that's what we have left. We are not going to cover or talk about that little snippet they have about mutation. But basically why we and how we know some of the stuff that we know at this level is being able to study cells that have mutations within these mechanisms, right? And they don't actually go into the greatest detail, and I don't think a level of understanding you guys would understand for that little section. So we're skipping over that section, which is the next one, and going right to eukaryotes. So that brings me to, when do we want to do our test? Going for a second. I'll pull up the calendar. So today, of course, is the 27th, right? We need to finish up Chapter 11 on probably November 1st. And then we're done with this module, right? And so we're ready to potentially take a test on it. I don't want to turn around Thursday and give you guys a test on it. I don't know that you guys would feel ready for that. No, I'm getting a lot of no's. Okay, your projects are due on the 8th, though. Go ahead, Michelle. I'm open to suggestions, because I want, it's not a dictatorship. Take the test on the 10th, or do we want to move the projects to the 10th and take the test on the 8th? Okay. Okay. We can do that. Okay. So, all right. I think most people who are here today get to vote. So, anyone in objection to the 8th and moving the projects to the 10th? Okay. All right. I didn't want to have 2 major things due the same day, so I will even post an announcement for who's not here today. Right. And I don't think they need to watch me